A healthy neural network operates with ruthless metabolic efficiency. Millions of action potentials fire simultaneously, demanding continuous, massive outputs of energy. But when that energy supply begins to stutter, the network goes dark. The resulting cognitive decline is a physical symptom of power failure. Alzheimer's, Parkinson's, and ALS present as distinct conditions defined by different protein aggregates. However, beneath these clinical differences, research suggests a shared vulnerability, a progressive failure in the organelle responsible for nearly all neuronal power. In this framework, bioenergetic collapse is a transdiagnostic driver of decline. The evidence for this commonality emerges from Russell Swerdlow's hybrid models, which isolate mitochondrial function from the rest of the cell. Swerdlow's work focuses on inherited maternal mitochondrial DNA, or mtDNA, haplogroups. These inherited mtDNA variants dictate the baseline efficiency of oxidative phosphorylation, the process of converting nutrients into ATP. This establishes a bioenergetic ceiling, an inherited maximum power output that varies between individuals. Age and environmental pathology erode this efficiency. Because everyone starts with a different inherited ceiling, the threshold of critical energy failure is reached at different times. To understand how this triggers decline, we have to map the physical collapse of the cellular energy engine from the inside out. The vulnerability of the mitochondrion begins at its perimeter, at a physical channel on the outer membrane called the TOM40 translocase. Mitochondria cannot maintain themselves independently. Over 99% of the proteins required to run the mitochondrial engine are encoded in the cell's nucleus and must be imported through the TOM40 gateway. This map of chromosome 19 illustrates a critical structural relationship. The gene that codes for this import channel, TOM40, sits in tight linkage disequilibrium with APOE, the primary genetic risk factor for Alzheimer's disease. A poly-T repeat polymorphism in TOM40 modifies disease risk by affecting the efficiency of the channel itself. If the TOM40 import channel degrades, the internal oxidative phosphorylation machinery cannot be maintained. The system reaches a point of failure because the replacement subunits required to repair the engine can no longer enter the organelle. This is the electron transport chain. Complexes I through V perform oxidative phosphorylation, passing electrons to generate ATP. This process is inherently volatile. At complexes I and III, natural electron leaks interact with oxygen to generate superoxide, a reactive oxygen species, or ROS. At low, transient levels, these reactive oxygen species serve as signaling molecules that trigger adaptive responses to strengthen the cell. But at chronic, high levels, excess ROS begins oxidizing local lipids like cardiolipin and damaging the mitochondrial DNA. Natural experiments illustrate the sensitivity of this chain. Environmental neurotoxins like annanasin and BMAA act as direct complex 1 inhibitors. They physically blockade the electron transport chain, halting ATP production and causing ROS to spike. When the membrane is sufficiently damaged by this oxidative stress, cytochrome C, which normally shuttles electrons safely, leaks into the cytoplasm. Once it breaches that boundary, cytochrome C activates the molecular machinery for programmed cell death. Reactive oxygen species are an unavoidable byproduct of energy generation. A neuron's survival is dictated by the ratio of this metabolic damage to the cell's capacity to repair and replace its mitochondrial engines. To maintain that repair capacity, the cell relies on NAD+. It is the essential electron carrier feeding the transport chain and the mandatory co-substrate for sirtuins, enzymes that regulate the creation of new, healthy mitochondria. This chart plots chronological age against the expression of an enzyme called CD38. As tissue ages and experiences inflammation, CD38 levels rise sharply. Simultaneously, the total cellular pool of NAD+ drops. Eduardo Cini's research identified CD38 as a primary NAD+ consumer in aging tissue. As its expression increases, it progressively drains the NAD+ pool across the cellular environment. This depletion creates a two-front failure. Without NAD+, the electron transport chain is starved of the electrons it needs to function. Simultaneously, the mitochondrial biogenesis program is deactivated because sirtuins lack their required co-substrate. This establishes a self-destructive loop. The cell is unable to manufacture replacement mitochondria at the exact moment its existing energy engines are failing. This is the mitochondria-associated membrane, or MAM. It is the physical contact zone where the endoplasmic reticulum tethers to the mitochondrion, governing the calcium transfer required to stimulate ATP production. When mitochondria are damaged, they undergo triage. Through fusion, they rescue mildly damaged partners. Through fission, they physically divide, segregating irreparably damaged segments. Those segregated, failing fragments are cleared via the PINK1-Parkin mitochondria pathway. This disposal system uses ubiquitin tagging to identify broken mitochondria and mark them for degradation. In neurodegenerative states, this contact site becomes a convergence point for pathology. Aggregation-prone proteins, specifically amyloid beta and alpha-synuclein, actively accumulate at the MAM interface. Their presence disrupts calcium flow and amplifies local ROS. In the mitochondrial matrix, amyloid beta binds directly to an enzyme called ABAD, further accelerating oxidative damage. While the initiating proteins differ, their impact on the organelle follows a convergent path. A-beta and alpha-synuclein both target the same mitochondrial triage and quality control systems, leading to a shared state of bioenergetic distress. When internal quality control fails, the damage spills into the cytoplasm, alerting the brain's microglial immune cells. Oxidized mitochondrial DNA escapes the failing organelle, activating the CGAS sting pathway and the NLRP3 inflammasome to output inflammatory cytokines. Escaping ROS also activates NFKB, driving amyloid precursor protein transcription. This generates more amyloid beta, which returns to mitochondria, causing further ROS, a self-sustaining cycle of escalating damage. The immune response carries a significant metabolic cost. Activated microglia and infiltrating T cells consume remaining NAD+ and glucose in the local tissue, deepening the energy deficit for the struggling neurons. This leads to the Panthos model of cellular backup. The waste disposal system relies on VATPase enzymes to acidify lysosomes and dissolve cellular debris. Because VATPase requires ATP, it fails when energy levels drop, causing uncleared autophasomes to pile up. Bioenergetic failure initiates a dual-front collapse. It activates an energy-draining immune response while simultaneously paralyzing the cell's ATP-dependent waste disposal system. To understand why certain cognitive functions fail first, we have to calculate the cost of this cellular collapse on brain function. The brain's energy demands are not distributed equally. Synaptic transmission, the firing of action potentials and the recycling of neurotransmitters, is the most energy-intensive process in the central nervous system. These fast-spiking GABAergic interneurons are the most metabolically demanding cells. They fire in sustained bursts at gamma frequencies, requiring massive, uninterrupted volumes of ATP. These cells must fire continuously to coordinate complex cognitive networks. This constant demand makes them particularly vulnerable to any interruption in the energy supply. Cognitive decline follows the rules of energy economics. When the bioenergetic ceiling lowers, these hyperactive GABAergic synapses are among the first to fail, explaining the selective circuit vulnerability observed in neurodegeneration. The ultimate test of the bioenergetic collapse thesis comes from directly treating the organelle. Methylene blue is used as a pharmacological probe to validate the exact location of the failure. Methylene blue functions as an alternate electron carrier. When complexes 1 and 3 are blockaded, methylene blue arcs over the damaged machinery and donates electrons directly to cytochrome C. Restoring the flow of electrons restarts ATP production. In preclinical models, this bypass provides significant neuroprotection, suggesting that the damaged mitochondrial engine is a primary rate-limiting variable in disease progression. This indicates that if the underlying bioenergetic supply has already collapsed, addressing downstream protein aggregates may be insufficient to restore function without also repairing the energy source. Disease modification likely requires restoring mitochondrial output,